Today's lecture will be on macromolecules. What is a macromolecule? A macromolecule are giant molecules called polymers. They may have a molecular weight of greater of 100,000 atomic mass units, and they are formed by covalent linkages of smaller units called monomers. Monomers bond to form polymers. The function of macromolecules are related to their shape and the chemical properties of their monomers. There are four major types of biological macromolecules, which are proteins, carbohydrates, lipids, and nucleic acids. For this presentation, we will focus on the macromolecule protein. But first, let me start out by explaining the macromolecule composition. All macromolecules are made up the same way in all living things, and are present in all organisms in roughly the same proportions. As you can see in this pie chart, it shows the percentages of proteins, nucleic acids, carbohydrates, and lipids, in which all living organisms are made up of. Now we'll be talking about a very special mechanism, which is dehydration synthesis, also known as condensation reactions. Dehydration synthesis is a mechanism by which macromolecules are formed. A hydroxyl from one monomer is linked to a hydrogen molecule from another monomer, and in this process, water will be formed. The reverse reaction in which polymers are broken back into monomers is called hydrolysis reaction. Here is a visual of dehydration synthesis. As you can tell, the hydroxyl from a monomer bonds with a hydrogen molecule to form polymers. In this slide, it is a vision of the reverse reaction in which polymers are broken into monomers, which is called hydrolysis reaction. Each different type of protein has a specific amino acid composition, shape, and function. The examples below are the most common protein functions, which are support in connective tissue, transport of hemoglobin, movement in muscles and muscle contractions, protection against disease like antibodies, and for enzymes, which are catabolic, or they speed up chemical reactions in your body. As I mentioned in the beginning of the lecture, polymers are made up of smaller units called monomers, and that's where proteins come in. Proteins are polymers of amino acids. So amino acids will be the monomers for proteins, which amino acids will form bonds with another amino acid, and will give you a protein. Amino acids are joined together in proteins by peptide bonds. As you can see from the figure, a peptide bond forms between the carboxyl group of one amino acid and the amino group of the adjacent amino acid. Now we will be talking about protein and the structural levels. There are 4 levels of protein structure, which are primary, secondary, tertiary, and quaternary. The primary structure refers to amino acid linear sequence of the polypeptide chain. The primary structure is held together by covalent bonds such as peptide bonds, which are made up during the process of protein biosynthesis. The secondary structure refers to highly regular local substructures. Two main types of secondary structure, the alpha helix and the beta strand or beta sheets, were suggested in 1951 by Linus Pauling and his colleagues. These secondary structures are defined by patterns of hydrogen bonds between the main chain peptide groups. The tertiary structure refers to the three-dimensional structure of a single, double, or triple bonded protein molecule. The alpha helixes and the beta plated sheets are folded into a compact globular structure. The folding is driven by the non-specific hydrophilic interactions, but the structure is stable only when the parts of a protein domain are locked into place by specific tertiary interactions, such as salt bridges, hydrogen bonds, and the tight packing of side chains and disulfide bonds. Quaternary structure is the three-dimensional structure of a multi-subunit protein and how the subunit fits together, such as hemoglobin, which has four subunits, 2 alpha and 2 beta chains. Now we'll be talking about denaturation of protein. It is the process in which proteins lose their tertiary structure and secondary structure by application of some external stressor compound, such as a strong acid or base, a concentrated inorganic salt, or organic solvent such as alcohol. It is also due to extreme temperature, pH, oxidation, or reduction conditions. And this process is usually non-reversible.